Good afternoon, and thank you for attending this afternoon's CumbriaMed Research Webinar. I'm Professor Jonathan Weaver. I'm the co-director of the Research Hub at the Pears Cumbria School of Medicine with my colleague, Professor Sophie Day. So the purpose of the webinars we run is to showcase research and clinical care from the new medical school in Cumbria, and also showcase the relationship between Imperial College and local collaborators and partners in Cumbria, both in the NHS Trust and at the University of Cumbria. So the webinars bring cutting-edge research on topical areas together with clinical presentations from the PCSM partners in Cumbria. And as this webinar, I think suitably topical given the weather, we're going to look at the science and medicine of sleep. So I'd like to introduce our first speaker, is Professor Nick Franks, who is a professor of Biophysics and Anaesthetics from Imperial College London. Nick's a scientist who's looked at the fundamental mechanisms of how general anesthesia actually works in the brain, and latterly has turned his attention to questions of sleep and the neuronal mechanisms behind that. He has an international reputation in his field. So Nick, thank you so much for coming along this afternoon. We look forward to your presentation.
Thank you, Jonathan. Thanks for the invitation. Okay, so yes, I'm going to give you a brief overview of what we know about how we sleep and why we sleep, and particularly what we don't know. This is my son here on the left and his little friend Rosa, who Jonathan knows. They're not as cute now as they used to be. They're both 47 years old now. But when I took this photograph, literally seconds before, they were protesting they were not sleepy. And within seconds, they were asleep. And that's one thing we know about sleep, is it really is inescapable. I'll come back to that point a bit later. Now, different animals seem to have different sleep needs. But you have to remember that when you're not sleeping, you're awake. And so it's easy to mistake sleep need for wake need. And the difference in the requirements and the length of time animals sleep have probably more to do with how long they have to spend awake. So an elephant, for example, and large herbivores sleep a very short amount of time, probably because they have to eat a non-nutritious diet and they have to eat to maintain a massive body weight. At the other extreme, small bats will sleep nearly 20 hours of the day. They have a highly nutritious diet that's plentifully available. So this has to do with how long they have to remain awake. We lie somewhere in between. Now, it's often said, scientists often claim that all animals sleep. That's as meaningless to me as saying all animals are conscious. We simply do not know why we sleep, and until we know that, we won't really know whether all animals do indeed sleep. But I think all mammals sleep. And the reason I'm confident about that is because we can measure the EEG signatures of sleep, in mammals that really are very closely related to what we see in humans. Now, the EEG is far from perfect in that it measures electrical activity on the surface of the brain and a thin layer, the cortex, where it records really only synchronous activity. The background noise, where there's a lot of activity going on in the brain, is not really recorded. It records only when the electrical activity sums up. So we're measuring only an essence of what the brain is doing during sleep, but it's an effective way, at least, of monitoring whether somebody is asleep or whether an animal is asleep, and perhaps how deeply they are asleep. Now, I won't go through this in detail. The only take-home message is the EEG is a signature. It reflects stages of sleep. During wakefulness, when the brain is doing a lot of work, it's largely a high-frequency noisy signal with little synchronous activity. But as the stages progress to deeper and deeper sleep, then the synchronous activity dominates, and you have these delta waves or slow waves, which are characteristic of what is thought of as deep sleep. Now, you'll all be familiar, I'm sure, with REM sleep, and the fact that during REM sleep, your muscles are paralyzed, and we understand the circuits behind that, that there's an activation in the brainstem of neurons that inhibit spinal motor neurons. Now, why you're paralyzed is not obvious. People say it's because you are prevented from acting out your dreams, but that's a little unclear because it is now been shown that the activated cortex, which occurs during REM, is not the real signature of dreaming. It measures REM, where you have rapid eye movement, and it's quite different to non-REM. But the dreaming, the characteristic of dreaming is, in fact, it has been shown a hotspot of activity in what's called high-density EEG. And at the back of the brain, between the parietal and occipital lobes, there's a hotspot of activity that is predictive of dreaming. And it does not correlate with paralysis. And so it turns out you spend a good deal of time at least having some conscious activity during non-REM, and that is somewhat mysterious. The characteristic of, if you ask somebody, are you dreaming? You wake them up, of course, you say, were you dreaming? And you get a different response than if you say, what's on your mind? In other words, you have conscious experience, but they don't necessarily correlate to the active dreaming state. Now, we do know how the brain triggers sleep. That during waking states, you have a large number of neuronal systems on the left that ascend up to the cortex, keeping you awake. But sleep is an active process. And if you look on the right diagram, from the hypothalamus predominantly, there are descending inhibitory signals that become activated during sleep, and these inhibit the arousal pathways. So sleep is a well-organized and defined active process that switches off these arousal pathways. And we did an experiment that illustrates this very clearly. You can genetically engineer mice these days by introducing genes, transgenes they're called, into specific neuronal subtypes, and they're introduced using a virus, adeno-associated virus. And we transduced the neurons in the descending inhibitory system such that when they became activated, they made a molecule which would respond to a chemical called CNO, clozapine N oxide, which would reactivate them days later. So these neurons retained a memory of having been activated during sleep. So what we did is sleep deprive the animals. Now, you all know sleep deprivation is a stressful business, and that's a confound in all experiments when you deprive an animal or a human of sleep. But with mice, you can do it quite gently. If you introduce novel objects into their cage, you can see here a sort of toy or a marble, that keeps them interested for hours. And you can prevent them sleeping for many hours. That's sleep deprivation on the right-hand side. And if you then let them rest, they immediately rebound into deep sleep for many hours. And during this time in these animals, the chemical that was transcribed by the gene that we put into the neurons is being synthesized. It has no effect normally on the animal, but if days later we give these animals CNO, which reactivates those particular neurons, the animals immediately fell asleep. So this tells you something quite profound, that just that local region of neurons was necessary and sufficient to induce sleep in these animals. And importantly, these are neurons that are activated. You think of that activation, of course, inhibits predominantly the brain, but is the process of sleep is an active process that triggers the activation of this inhibitory response. So that's message number one. Now, that's predominantly what we know. I'm now going to move on to what we don't know. We know, of course, sleep is essential. By the time you're my age, or Jonathan's age, you would have slept 20 years or more. That's a colossal amount of time, and it must have some fundamentally important process to risk being asleep for that length of time. For some animals, sleep deprivation can lead to death faster than food deprivation. So it's clearly absolutely essential. And Alan Rechtstaffen made a very cogent point, but that if sleep does not serve an absolutely vital function, it's the greatest mistake evolution ever made. Clearly, to move into a state of vulnerable inactivity and risk predation, you must be doing something pretty important. But we still don't know what that is. That remains the biggest mystery of sleep, is what is the goodness of sleep? We can measure whether you sleep, and you'll all know if you wake up after what seems like a long night's sleep, but you haven't been satisfied with that sleep. We don't know what that is that you're missing. I think the most striking illustration that sleep is essential, cetaceans like this beluga whale have to move from birth to death. They're constantly on the move because to get enough oxygen. So how can they sleep? Well, the trick that they employ, and so do geese that fly very long distances over days, is half of their brain goes to sleep and the other half stays awake, and then it flips over to the other side. So they have hemispherical sleep. So you can see the EEG trace on the bottom oscillates between the left being awake and then the left hemisphere falls asleep, and the right does the opposite. So this is a beautiful example of how evolution, even in animals that still have to move around a lot, have solved the problem of sleep. But what is it that they get by sleeping? Well, all sorts of things go wrong if you don't sleep, and I won't list them all, and they may not all, of course, be the reason you have to sleep. But here's an important one, a classic experiment in which humans were given a hand-eye coordination test, and it was measured, this panel on the left, with increasing hours of wakefulness, in other words, sleep deprivation. And there's a decrement. It's not huge, it's maybe 7% after 24 hours of being awake. But that decrement is equivalent to, if you look on the right-hand side, you get the same decrement with the level of alcohol in your blood at the drink driving limit. So this is a significant decrement. There must be some degradation in some reflex pathway, and that, even in evolutionary terms, is quite sufficient to give you an edge over your predators. So it may be as simple as that. And one thing that people worry about a lot, particularly as you get older, is that there is some evidence, it's not overwhelming, but some evidence that poor sleep predisposes you to dementias like Alzheimer's. And it's based on this. Firstly, there's statistical evidence, but mechanistically, it has been shown that the molecules that form plaques in the brains of Alzheimer's patients, amyloid beta, does have higher concentrations in the waking state and lower concentrations during sleep. And the problem here is back to this zero-sum game of waking and sleeping. Amyloid beta is produced more during the waking state. And so we do not know whether the reduction during sleep is due to removal being more effective, which is what the argument is, or simply that less is being made. Now, the fact that clearance may be important during sleep has given rise to probably the most dominant theory of sleep in the last decade. I won't go through this in detail, but the principle is simple enough that a group in Rochester seemed to show that the clearance of amyloid beta and other metabolites and toxins were cleared faster during sleep. Now, they showed, in fact, that entry into the brain was faster during sleep, and they came up with what many of you may have heard of, the glymphatic hypothesis that posits that that is part of a clearance system. Now, unfortunately, we have thrown a spanner in the works in that particular field because we find the exact opposite result if you put metabolites into the brain and measure how quickly they're cleared. In fact, they're cleared less well during sleep and during sedation. So that remains a contentious area. And in my last slide, another very popular theory that I think is more likely to be right than any other. It has a fancy name like synaptic downscaling, but what it is basically is during the waking state, the brain is extremely active and synapses are being formed and being strengthened. Some synapses might be weakened, but many are strengthened. And the argument is that during sleep, there's a sort of downscaling, a pruning of synaptic strengths to reset the system. And if there's one message I'm going to leave you with about theories of sleep, it's this. If somebody says this is the reason you sleep, say memory is consolidated better, which is a popular idea, you just ask the question, why can't it happen during the day? And there has to be a good reason. The brain has to be unconscious and you're rendered invulnerable for the mechanism to be relevant and necessary during sleep. So I'm going to end there, I think just about almost on time, and hand over to the chair, Jonathan.
Nick, thank you so much for that very elegant presentation. And I look forward to a discussion about it. We're going to go on to the second speaker, and we'll take the questions and the Q&A after both speakers have presented. I can ask you to, perfect, thank you. So I'd introduce the second speaker this afternoon is Professor Mary Morrell. Mary will be well known to many in Cumbria as the head of the Pears Cumbria School of Medicine, the foundation head. And Mary is a nurse by background and a professor of sleep and respiratory physiology at Imperial College, and as well as head of the Pears Cumbria School of Medicine. So she's an extremely experienced sleep investigator and with particular experience of working with sleep apnea. So Mary, look forward to hearing your presentation. Thank you.
Thank you very much, Jonathan. I'm just going to see if I can share my slides. So I'm going to share. Well, thank you very much, Jonathan, for that intro. Sometimes it's interesting for people to see where you originate from. So with great trepidation, I'll show you. We don't appear to be working you in terms of. There we go. So I thought I might show you that I went from nurse to doctor, and then have moved up to Cumbria as the head of the Pears Cumbria School of Medicine. But while I was doing my research, I kind of fell into sleep really from a nursing background. I took a job with a professor who was very interested in how breathing worked, and he really wanted to know why people, if they were breathless when they were awake, weren't breathless when they went to sleep. And this is my favorite slide of all time. On the left-hand side of this slide, you can see somebody who is deep asleep. The EEG, which Nick was talking about, is sleep-related. And you can see the respiratory effort is quite regular. And then you can see something that says arousal. So I may have just shut the door or dropped my pencil or something and woke the person up. And you can see it's tipped them into respiratory instability. So they get a crescendo, decrescendo, hyperventilation, rapid breathing, and then no breathing. And I think it's amazing that a human being can breathe regularly and then get tipped into this respiratory instability. And what causes that and why does it happen? So that's what I got involved with studying. And of course, it's quite complicated studying humans because you can't always do the experiments that Nick can do. So in humans, you've got two ways of controlling breathing. One is with your motor cortex, where you can control your breathing, so you can sing and play wind instruments, etc. But obviously, when you go to sleep, your motor cortex is shut off, as Nick's indicated, and you're breathing mainly with your brain stem, and therefore you're using your bulbospinal pathway to breathe. And obviously that sleep intervention is the thing that we were trying to study. And when you don't have animal models, sometimes you have to use humans where they've got bleeds in certain arteries. This one here is the lateral medullary syndrome that knocks out half the brainstem. And you can use these human lesion models to try and understand the control of breathing. So, as I said, there's periods, there are some people who don't breathe when they go to sleep, and this is called sleep apnea, with apnea being the cessation of airflow. So in central sleep apnea, your brain's not generating any respiratory effort when you go to sleep, and that's related to your control of carbon dioxide and oxygen when you go to sleep. Fortunately, that's quite rare. But the other sort of sleep apnea that some of you might have heard of, which is obstructive sleep apnea, is quite common. And in obstructive sleep apnea, the area at the back of the throat and at the soft palate blocks off, so that you're trying to breathe, you're making respiratory effort, as you can see on this slide here, but you've got no airflow. So there's a problem between the control of your respiratory centers and the control of your respiratory muscles. And for a while, I spent time in America, and I was putting pediatric fiber optic bronchoscopes down into the back of the throat to try and measure breathing airway collapse in real time. And if this works, you'll be able to see somebody's airway collapsing in real time. The white thing is a pressure catheter down there, but you can see every time this person breathes in, out, in, out, the airway's collapsing off because it's getting blocked off. We looked at all sorts of different people, and turns out different people block their airways in different ways, but the result is the same. The airway gets blocked off. So that's a problem. Why is it a problem? What are we going to do with that information? Well, if you look at the cycle of obstructive sleep apnea, what happens is you fall asleep, you get a decrease in tone to the upper airway, which when you generate negative pressure in the chest to try and get air into the lungs, it causes the airway to block off. You get a cessation of breathing, apnea, and that causes a dip in the oxygen. But then to wake yourself up, because you've got a dip in oxygen, you've got an increase in carbon dioxide, you're making respiratory effort, your brain knows you're supposed to breathe in obstructive sleep apnea. You wake up from sleep. So the symptoms of sleep apnea are dips in oxygen and fragmented sleep. And as Nick's already told you, sleep is really important for the brain. It's likely that the intermittent hypoxia has cardiovascular effects, but the lack of sleep and the intermittent hypoxia is probably affecting the brain. So one of the things that I then did is I went on and tried to find out what might be happening in humans where I can't do the elegant experiments that Nick was doing. So at that time, we decided that we'd do a bit of brain imaging, and this was when magnetic resonance imaging was really beginning to come into its fore. And this one over here with these 7 subjects is one of my most highly cited papers because it's the first time that people showed discrete hippocampal regional damage in OSA subjects, some people with sleep apnea. And this is really speaking to, does the intermittent hypoxia, does the brain imaging affect the brain? And obviously we went on and tried to explore that finding in more people. But the reality is that this research is messy. You can't do the studies that Nick can do in humans. And so Nick's already talked to us about Tononi's theory that why sleep might be happening. And we've written some discussions about why and what might be happening to the brain. But the fundamental problem that we have in humans is it's very difficult to do these controlled experiments to explain what might be happening in people who have fragmented sleep. And part of the reason for that is that it's really difficult to do the experiments in humans. On the left is one of my subjects. He loved doing this modeling picture for you. And we tried at some point to try and create better measurement techniques. So this is some work that I did with the engineering group where we tried to put some of these sensors into an earplug into your ear to try and improve the measurements. This didn't quite work, but I was lucky enough to be involved with a really excellent European bid. And what they did is took some of these measurement techniques and they made them into a little sensor that goes on the chin that can measure not sleep, but the respiratory effort during sleep. So it's really important that we look at the technology that we use to try and improve the measurements that we're making. And I just want to say that this is an important issue. I mean, we all sleep, as Nick says, but depending on which way you count it, there may be up to 1.5 million people in the UK with some kind of sleep apnea, either severe or mild sleep apnea. So this is another paper that we did trying to estimate the prevalence of sleep apnea across the globe. And one of the things that causes the collapse of the upper airway is if you have adipose tissue in the upper airway, and it is a disease that is highly prevalent. Another thing that's interesting about the disease is it's easily treatable. You can treat it by blowing air into the upper airway with a little machine that I've shown here. And provided you wear a mask on your face or on your nose, you can blow the air into the airway while you're asleep and you can make it work. But not everybody finds it easy to use those machines. And one of the interesting things, if you're thinking about, well, who should I give the treatment to? What kind of treatment should we be giving? Is that one of the things that I wanted to flag is that when you get older, there are certain things that happen. And one of them is that your hyoid bone moves forward and down. So sleep apnea is actually quite common in older people. So that's quite interesting because if sleep is needed for some of the reasons that Nick was saying, and if we've shown that in sleep apnea there is some neural damage, then it may be that sleep apnea is a modifiable, treatable thing in older people that might be linked to cognitive function. So we were quite interested in that. And one of the things that we then did, having developed this idea, is look at, well, should we treat older people with sleep apnea and does it make them feel better? So we networked a number of the sleep labs together in the UK, and we did some quite large studies looking at, particularly if you treat older people with sleep apnea, do you make them feel better? It's typical, isn't it, to say, well, older people, they can sleep more and they don't have to do quite as much work, so, maybe it's not a problem. But actually, if you treat older people with sleep apnea, it does make them feel much better. And interestingly, when we're doing studies that are funded by NIHR, we always need to do cost effectiveness. And it does turn out to be cost effective if you construct the relative economic models to say that it is worth treating older people with sleep apnea. So then we asked ourselves if it is okay to treat older people who might not have such severe sleep apnea, should we treat people earlier? And if we're going to do that, what's the cost implications of treating more people? You have excellent sleep services, both in North and South Cumbria. And if I'm going to increase the number of patients that they are going to see, they're jolly well going to want to see the evidence for it. So the second study that we did was actually funded by industry, and that throws up some interesting questions. But we networked again the sleep labs in the UK together, and we now use technology to try and improve people's adherence to treatment. And we actually showed that if you do this study and you treat milder sleep apnea, you can actually get good results. And these data fed into the national guidelines for the treatment of sleep apnea. So by taking part in these studies, as people did throughout the UK, they contributed to changing guidelines, which I think is showing the value of research in the way that it influences treatment at the highest level. So then we just briefly, because Jonathan will be ready for chopping me off now, we briefly looked at the adherence to treatment. Not everybody wants to use this treatment, and technology is moving all the time in how we measure sleep, how we can treat sleep. And most of you will be using your phone. You'll have, you might have rings, monitors that measure what they say they're measuring indexes of sleep. So the most recent trial that we did was a study of an alternative treatment for sleep apnea. And this was actually a research for patient benefit study run by a physio colleague of mine. And it was run in conjunction with the Sleep Apnea Trust, looking at positional sleep apnea and whether you can improve it by using some of these devices. And I think this is a really nice model of how we can do studies going forward. So my take-home points are that sleep apnea is caused by changes in control of breathing. It's common. It causes arousal from sleep and dips in oxygen, and we need research to understand the implications of that. The research that we do can often support treatment decisions, and that has particularly led in the area of sleep to recommendations around treating sleep in older people and milder sleep apnea. But if we are going to put forward alternative treatments, I do think that they need to be evaluated appropriately, and particularly if that leads to an increase in patients that we might want to treat. So thank you very much for listening to me, and look forward to questions.
Thank you. Thank you, Mary. I'd like to thank both the speakers, Nick Franks and Mary Morrell, most sincerely for a really fascinating webinar. Can I thank the audience for all their contributions and questions? And I hope we've all learned a bit more about sleep. Once again, many thanks to both speakers. Thank you.